Okay. Well, as I already, okay, so maybe I should introduce myself again. My name is Martin Eimer, and I'm a professor here at Birkbeck. And as I have already mentioned this morning, the reason why we're here at Birkbeck is because the ESRC has funded a specific research project on prosopagnosia, and allowed us to have this little open day here. And what I would like to do in the next sort of 20 minutes or so is just give you some idea what we're actually doing in this specific research project. And since I see some of you here who have actually participated as experimental subjects, it might be quite interesting for you to get some idea as to what's the thinking behind it. So it's basically looking at face recognition in face blindness with brain activity measures or event-related brain potentials. Maybe I should say first of all that I actually come to face blindness from a slightly different direction. I mean, unlike Brad, I haven't actually started out being interested in face blindness. I started out being interested in face processing in normal, in how you and me, or me and me. So how does face processing work in people who don't categorize themselves in face recognition? And that's difficult enough. But then, of course, if you're interested in normal operation, it's sometimes really instructive to study people that have some selective deficits. So it's interesting also from a sort of general perspective. If you're interested in normal face processing, it's actually quite interesting to study people who, well, something wrong. And maybe we can actually then refer back to normal face processing. So it goes both ways. So basically what we're doing in our research is to understand, well, we're trying to understand how cognitive mechanisms work in the brain. And what we're doing in our research is, and that's some of you who have been participants will remember, we are basically recording electrical brain activity. So we have participants here sitting in front of computers watching stimuli like faces. I'm actually wondering whether, can you see in the back, or would it be, okay, that's great because we have some light here. So we are basically looking at the brain response to faces and other stimuli, and we measure the brain response with EEG. So basically we are putting electrodes on people's heads and measuring the brain response. And before I tell you a little bit about the kind of questions that we ask and the kind of results that we got, I'll just give you sort of some brief guided tour through the EEG lab, just in sort of a few slides. So if you were a participant, or some of you have been a participant in our EEG lab, what would you actually experience in terms of pure practicality? So basically you would come in and then you would have sort of nice research assistants who would basically, first of all, measure your head, because we need to know exactly which electrodes go into which place, and each head is different. So we're basically looking at the circumference of the head to find the right electrode caps, because that's the next thing that happens. So we put these electrode caps on your head, which already have electrodes in pre-specified positions, and of course we have to find the right size for you. So that's the first thing. Then the electrode is put on. It's all perfectly painless, and although it doesn't look like that, but it is actually. So electrode is put on, sort of strapped tightly, and then of course in order to really record the electrical activity, we need to get as good a contact as possible. So what we're doing is here with these little pads, we're sort of rubbing a little bit of, or using some alcohol to basically clean your skin and get as good a contact as possible from for different electrodes. And there you see this. So normally it's actually not 3 people here. I think normally it's only one. We did this for effect, or if it has to go really fast, but normally it's just one person who's sort of working on your head at any given time, and it's not painful. And then afterwards, that looks truly horrid, but it's actually just filling electrical gel sort of into the electrodes. It doesn't go into the head. It just goes into the hole in the electrode and sort of makes contact with the skin. So that makes it possible for us to actually record the good signal here, and then we're actually measuring afterwards whether the signal is good. There's an impression from our old lab with washing machine and washing powder, so it's very high tech here. So that's what we're doing, and then afterwards, of course, everybody's happy, and then participants are connected to the amplifier prior to recording. And the most important thing is electricity goes one way. So many people really are worried that we might actually reverse the direction of electricity and send electricity through the electrodes into your brain. That is not possible. That's not what we want to do, and that's not what we physically can do. So what we're doing, we're using these amplifiers to amplify electrical activity that is measured from your brain. So it's exclusively one way, perfectly safe. That's what we're doing. Okay, so, and then obviously we have faces and other stimuli and all kinds of interesting experiments, like for example, we ask people to identify a face, to tell us whether they recognize this face, find it merely familiar or completely unfamiliar, and then people are standing outside looking at the EEG activity, sort of. But basically, and I'm not going to make this very technically here, but basically the problem with the EEG recording, I mean, you all have seen sort of in clinical context, sort of when people when neurologists measure the EEG, for example, for diagnosis, when maybe there's sort of a question whether or not somebody is epileptic, or for any other reasons. The EEG is a sort of very strong signal that you can see easily. The problem with this is that if we're interested in face processing or any other cognitive processing, the EEG that we sort of see when we're recording from electrodes is really an extremely noisy signal. It's like a radio that is not tuned properly. So there's a little bit of signal there in the background, but overlaid in front of this signal is lots and lots of noise. So basically, if we want to see how your brain operates or processes faces, we look at 95% noise and we can hardly see anything. And that's why we do these strange experiments that lots of you have experienced, where we actually present faces over and over and over again. What we're doing then is a technique called averaging. We don't have to go into any detail, but basically it means that we improve the signal. So we're basically tuning the radio so that we can then actually see your brain response specifically to a given face, to say, for example, to a famous face. And that's what we're doing during our experiments. And obviously this is then the question, how are faces processed by the brain is what we're really interested in. And actually the logic of our experiments is embarrassingly simple. We are always looking at contrast. So for example, if we're interested in the most obvious question, how and at what point does the brain distinguish faces and non-face stimuli? Then we present faces, and these are usually faces of unfamiliar people like this one, and non-face stimuli like houses, and then we just measure the brain activity, sort of tune the brain so that we can actually see what happens. And then we ask the question, when can we pick up a difference? When does the brain start to discriminate between faces and non-face stimuli? And not just when, but also where. And then we have these sort of typical ERP responses, and this morning Sarah showed an example here. This is the N170 component. So, well, basically, what you have here is the brain response to a face, that's the red line, and to a house, that's the yellow line. And what you can see here, initially, there's no difference at all. The brain doesn't care about whether this is a face or a house, it's just a visual stimulus. But then at about 150 milliseconds, that's about a little bit longer than 1/10 of a second, so it's extremely rapid. There's a difference, we can see the difference. There's a sort of much stronger response to faces as compared to houses, and we have this similar picture this morning. So that's the N170. That's a differential brain response to faces as compared to houses. And that's really early, 150 milliseconds. And it happens in brain areas that are sort of close to the fusiform face area. So we believe this has something to do with face perception. But then, of course, there are other face-related questions. So, for example, differences between emotional and neutral faces. So one obvious important thing about faces is that they convey emotional information, whether somebody who's looking at you is angry at you or not. Extremely relevant. And we can then also use the same simple technique. We either present neutral face, or in this case, this is excellent, anxious face, fearful face, and then we see again, when does the brain pick up the emotional expression, that there's something in front of you, not just a face, but a fearful face. And what we can see is, well, again, very early, within about 150 milliseconds, we have a sort of differential brain response here. So the fearful here is this big peak here at about 150 milliseconds. And Actually, well, if you look here, what you see here, this is the back of the brain. This is where the visual processing happens. Where we get the fear response is somewhere different. It's up here. So that's very surprising. So there seems to be different areas. And then, of course, we can ask the question that maybe is most important for research on prosopagnosia. That is the difference between a famous and a non-famous face, or a known face and an unknown face. So what we often do is not present faces of personally familiar people like relatives, but actually famous faces, or faces of which we believe that they are famous. And like, for example, Einstein here. So if you have Einstein and a non-famous face, where does the difference happen? And how long does it take? And again, we see a difference in brain responses to famous or familiar and unfamiliar faces. This time it's slightly later. So the first brain response to faces as compared to houses, we get at about 150 milliseconds. And then it takes about 1/10 of a second before the difference between a famous face and a non-famous face starts to emerge. So there's definitely a sequence. First, you identify something as a face, and then you actually identify the stimulus, or sort of recognize the stimulus as a familiar face, which sort of intuitively makes sense that one has to come after the other. And this is the N250. So this terminology, if there's a number like 250, that basically always means that's how long it takes before you can pick it up. 250 milliseconds, that's 1/4 of a second before the first indication that the brain has started to recognize a face has happened. So this is all stuff that we have done with normal control participants, but of course we can now use these markers for different aspects of face processing and ask the question, in developmental prosopagnosia, which of these stages are affected? Maybe delayed or maybe completely wiped out. So what we can do is, for example, look at the N170 component. That's the first one. The N170 component is very early, and we can actually look, when we just have lots of electrodes, we can look at sort of this distribution of electrical activity all over the scalp surface, and we can then use that to actually have some guess as to which part of the brain is responsible for this component. So in this case here, I've just sort of indicated that we have good reason to believe that this N170 component is generated somewhere in the vicinity of the fusiform face area that Brad talked about. So ERPs and face blindness. We can, for example, ask the question...
Excuse me.
Yes?
I'm a bit stupid, I think, but why have you got some going up and some going down? What's the difference? I can't quite understand what the difference is between the bits that are going up, the lines that are going up,
okay.
the lines that are going down.
So what does this... Okay, so I should have explained this a bit.
Just explain that bit.
That's right. Okay, so first of all, this is time. So at this point here, at 0, this is when the face is presented.
Yes.
Yeah. Okay, and then we have electrical activity, and electrical activity measured with EEG is always recorded from electrodes, and that can be positive or negative.
Right.
And the up and down only refers to whether what we measure is a positive or a negative response, so electrically positive or electrically negative. There used to be a time, God, where's the mouse? All right. There used to be a time when people thought it's really important whether it goes up or down. People believe that up means activation, down means relaxation. We don't believe that anymore. The positive or negative that we measure in terms of brain activity really only depends on the orientation of the electrode. Has nothing to do, nothing functional to do. So what we're really only interested in is differences. So going up a lot means not there is a sort of a lot more activity necessarily. It's only the difference. But sorry, I should have said this. Say this again. So forget about the last. Delete the last 10 seconds. Up and down in itself, whether it's up or down, doesn't make a difference to whether it's more active or not. But how high it goes up or down does make a difference. So in that sense, here, for example, we have faces elicit a larger, higher response than, in this case, houses. Then it could have been the other way around. It could have gone down as well and be a bigger response. But positive or negative doesn't make a difference, but the size of these components does make a difference. Okay. So, here's face perception. Okay, now this is the Birkbeck voodoo again. Okay, there we are. Then we have face recognition, and then we have emotion detection. So what we are doing in our research here at Birkbeck is basically seeing which of these component processes are selectively affected in developmental prosopagnosia. And that's why we invite you, or those of you who have already participated, in doing these experiments. So the first thing, obviously, that we want to know is there actually some problem in really early face perception, the N170 components. So we look at the N170 component, and we basically just look at the difference in the brain response to faces versus houses. And what we find is that actually it's all right. In most developmental prosopagnosics that we've tested, what we do statistically is we look at each individual participant and then say, is there a reliable, statistically reliable difference between faces and houses? And what we find is that in 8 of the DPs that we tested, it just looks absolutely normal, strong N170 component, indicating that sort of the initial stages of face processing is intact. Then we have 7 so far that where the N170 isn't statistically reliable, but if you look at this sort of black is face, red is house, there's still something there. So the basic idea here is that face perception as measured by this N170 component is actually not too bad in most people that we've tested so far with developmental prosopagnosia. And that's actually quite different from acquired prosopagnosia. So about 13 years ago, when I was in Cambridge, I tested acquired prosopagnosic patient and published a few papers. And this patient was specific because he had no N170 whatsoever, and there are 1 or 2 other patients in the literature. So it seems as if in developmental prosopagnosia, face perception, as measured by this early brain response, is not selectively affected. Now, what we're really interested in is face recognition. So what we have done with our participants, both with controls and with DPs, is to let them identify famous and non-famous faces. So these are the stimuli that some of you may sort of remember vaguely. So we have a set of famous faces, I think it was a total of 80, and non-famous faces. On every trial, we ask our participants, now tell me whether you recognize this face. And actually what they have to then say is absolutely certain, pretty certain that it's not a famous face, or absolutely certain that it's not a famous face. So it's a sort of 4-way discrimination. Now, if we do this in our normal control group, more than 80% of the famous faces are recognized. There are faces like this here, sort of there's George Bush, and there are pretty famous faces in the UK. And when we then look at the difference between the brain response to the recognized famous faces and the non-famous faces, we do find, which I've already shown, that at about 250 milliseconds there is this N250 component, which sits here at the back of the head, and which we believe is something like visual face memory. So the idea is there's a perception of a face, there is a specific face that comes in, but in order to recognize the face, of course, what you need to do is you need to compare this face to some sort of visual memory of a specific face. In other words, in order to recognize George Bush, you have to have some representation of how George Bush looks visually. And what we believe is that this early component is actually the successful match between the incoming face and a visual face representation. And that is indeed what we can measure in our normal participants. And then later, much later, at about half a second, we have another brain response, which we call P600, which we believe has something to do with a real conscious recognition. This is George Bush. Not just this is a familiar face, but this is George Bush. Now, what about the DP group? Now, this is where you come in, because these are now the results from our recent experiments. Well, first of all, not surprisingly, we're using exactly the same very famous individuals. We find that in our DP group, less than 30% of these faces are recognized in the sense of classified as definitely or potentially familiar. Well, that's what we would have expected. What came as a surprise for us then was that when we looked at famous faces that were classified as unfamiliar, as non-famous, that the same N250 component that I've just described as face memory was actually still there. So, but I should qualify this. These are the data from exactly half of the participants that we tested. So in half of the DPs tested, famous faces that were declared to be unfamiliar did elicit this memory component. In other words, it looks as if the information from the face not just did get in there, but actually was successfully matched with some stored representation of a famous individual. What was completely missing was the later component, this P600 component, in the same individuals. So no conscious recognition, which actually corresponds to what you told us. You told us, I do not recognize this face. So in other words, what we see in half of our tested participants is that visual face memory appears to be intact for famous individuals. And this is a phenomenon that has been described in the face processing literature as covert face recognition. Sort of subjectively, consciously, the face seems unfamiliar, and that is what we report. But still, some part in your brain, sort of some early response in the brain related to visual face memory still distinguishes between famous and non-famous faces. Now, it's important to say this was exactly the case in 50% of all the DPs, of all the prosopagnosic people that we tested. In the other 50%, there was nothing. So as if this visual face memory was either not present at all, never formed, or people were unable to access it through the faces that they saw. And of course, for those of you who have participated in this study, Angie is here and will be able to tell you whether you have the face memory or not, because we know exactly on which side you can actually have this sort of nice discrimination. Okay, so I should come to an end anyway. Face recognition. So it looks as if visual face memory is preserved in some, but not all people with developmental prosopagnosia, which we call covert recognition. So then, of course, well, if there's a face memory there, so why don't they recognize the face when it's there? Well, something must be broken. There must be something maybe like a broken link between face memory and subsequent conscious recognition. So there's some spark there. This face matches a face memory, but then something else. It's sort of further down the line where information about identity is stored. This cannot be accessed. And of course, this is something that we would like to follow up. So future directions, and we have already got the data, we are currently analyzing them. The question is emotional facial expression. That's the third process that I haven't talked about here. Can people with DP detect emotional facial expression in exactly the same way and very rapidly as normal participants do? And as it seems our initial data say, it depends. Some can and some can't. This seems to be the general answer that we get from these studies. Developmental prosopagnosia is a really heterogeneous phenomenon. Problems can arise at different stages, and just because you have difficulties recognizing famous faces doesn't mean that you all have exactly the same problem. Some people have early face perception problems, some have later problems, some have face memory deficits, some have preserved face memory, and identification processes are affected. And then, of course, the question is, if we have face perception, face recognition, and emotional face processing as possible processes that can be affected in developmental prosopagnosia, how are these deficits linked? Can you have one without the other, or do they all go together? And then finally, and that's what Sarah also mentioned, is can we fix what we presume, the broken link between face memory and recognition through training? That's something that we have just started thinking about. I mean, the basic idea is that if there are, at least in some people, the brain recognizes a familiar face when the person does not consciously recognize the familiar face. Is there a possibility for us to actually provide feedback and say, look, something there, the brain is active, and we sort of provide some external clue, we could call this biofeedback, some sort of beep that says, okay, this was a familiar face. Is there something that you can actually access and maybe help you to sort of in the future sort of make this discrimination that at least consciously is not possible at the moment? So, but that's for the future. Can we fix this broken link between face memory and recognition through some sort of feedback training? Okay, I think that's all that we have time for at the moment, and as I said, Angie will be happy to give you some more detailed information. We have just got a research paper of our first findings related to this covert recognition submitted to a scientific journal where we also got very good reviews. The reviewers then asked one question, and that was a very good one. That was, well, you showed them famous faces, but how can you be sure that they actually knew who this person was? Did they actually have a concept of, say, George Bush or something like that? And of course, we didn't actually ask you afterwards. Do you know who George Bush as a written name is? So what we're going to do, what Angie and Joanna are going to do to all of you who have participated in this study, you'll actually get an email from us with a list of names, and we want you to actually tell us whether you know these individuals. Not from their faces, but actually from their written names, because of course we hope very much that you know who George Bush is, because that will help us in our argument. Anyway, thanks for listening. So, question?
You probably do know, but just in case, in the acquired literature, there's a similar dissociation in the covert recognition, because when I did the work with David, and he thought I was George Michael, we were trying to, I assumed that all acquired prosopagnosia would show covert recognition. So I was really disappointed that he didn't show the covert thing. And then when I read one of Andy Young's papers, I found out that actually only some of them show the covert, and it depends on at what stage in the whole functional model something's gone wrong. So I suppose in a way that's what your story is going to develop into, that these two groups are showing a problem at a different level of the system.
That's right. There are a few people with acquired prosopagnosia who show covert recognition, but it seems that it's more rare in the covert case. So in our case, we have 50%, which is surprisingly high. In sort of prosopagnosia with brain damage, it would be much lower. Normally, there's less evidence for that. Yeah.
What you were saying about sending out the info about people's names and do you recognize so and so? There are a few people with prosopagnosia who also struggle greatly with names.
Perhaps if you also put a quick descriptor beside the names, like, I don't know who so-and-so is, but I know who the actor on the house is. That I have an awareness there, although the name wouldn't register for me.
Yeah, we probably have to be. I mean, we'll be working against our own hypothesis, but so in favor of our own hypothesis. I mean, identify famous people. So here this guy is an American president. Okay, well, famous? Yes, absolutely. So we have to be a bit more subtle than that, but you're absolutely right. We have to think about how to do that. Okay, 2 more questions.
On your task, is it a familiarity decision task? You're showing the face and
that's right, we have half famous, non-famous faces, you make this 4-way distinction.
So if you just guess randomly, you've got a 50% chance of being right.
Absolutely. Absolutely.
That's
why we have to, that's why we use P prime, of course. We have to see, obviously what we want is, with our participants, that for non-famous faces, they consistently say no. So absolutely, and that's what we actually do find. So in the case that 30% for the prosopagnosis, they identified 30% of the famous faces as famous, but almost 100% of the non-famous faces as unfamiliar. So we're reasonably sure that they make this discrimination properly.
And on the prosopagnosis who show covert recognition, there's a technique that's been used in the acquired literature. So if you find sort of Bruce and Young view that that covert recognition is sub-threshold activation, something like this, it's just not enough to sort of get through into consciousness. What the technique that we've used within acquired prosopagnosia is to show a group of famous faces all from the same category. So you show, say, 6 actors. So the idea is they all produce activation up to the semantic information about actors, and then the patient could then name the faces because they were mutually supporting each other.
So it means that the 4th and 5th and 6th would be more likely to be named if it was in the series.
Yeah, so no, you show them simultaneously.
Oh, I see. Yeah.
And so beginning they say, no, I don't know these faces. They say, oh, hang on, I think that might be so-and-so. And then gradually It builds up on each one.
That's recognition by context. That's a coping strategy.
Yeah, it is a form of context. Yeah. So that works with some, and I've actually seen it. We had an acquired prosopagnosia who went from complete chance performance to normal performance with those groups. So it might be one technique to explore.
Yeah, absolutely. Probably the ERPs is always you want to have one stimulus at a time. So that would be a behavior.
The ERPs on it.
That's right. Okay, that was one.
When the prosopagnosia actually identified famous faces, does the P600 similar to them as in normal?
All right, on those few trials where they did identify famous faces, indeed, very good point, they also have the conscious recollection component. So when, on those 1/3 of all trials where they identify faces, basically look normal. Yeah. Which also is interesting because there are some people who say developmental prosopagnosia means that the brain processes have been completely reorganized, because from very early on you were actually not able to sort of processing face identity in a normal way. So maybe it all looks really completely different. That's not what we're seeing. We're seeing exactly the same processes going on, only less likely to happen. But when it works, it seems to work normally. Okay, since Joe isn't here, we might.
Okay. The issue of the heterogeneity within the condition, and I just wanted to point out it's probably even slightly more complicated, because even within perceptual processes there will be differences in the extent to which people can rely on individual face features, for example, versus the global configuration, or even different types of configural processing. So, it is more complicated than it looks, even though it has a single name.
Absolutely, that's right. You have to start somewhere. Okay, thanks. So now I think we'll be ready for Joe.